Good morning. My name is Dr. Laura Bachman, and I'm the Chief Medical Officer and the Clinical Team Lead in the Program Development and Quality Improvement Branch for the Division of STD Prevention at the Centers for Disease Control and Prevention. I want to welcome you to the second day of the 2020 CDC STD Prevention Conference. Before I introduce this session, I wanted to remind you that if you're interested in continuing education credits for this session, please visit the website for additional information. Also, in the event that we have time at the end of this session to answer questions, please enter your questions for the speakers in the Q&A box on the right hand of your screen. It is my pleasure to welcome you today to the plenary entitled A PEP Rally for Biomedical STI Prevention. Today, we will hear from two international experts in the field on two cutting-edge issues. First, Dr. Kellum will discuss pre- and post-exposure prophylaxis for STIs, promising interventions to tamp down rising STI rates, but also controversial topics in the era of antibiotic stewardship and the growing concerns about antimicrobial resistance. Dr. Kellum will be followed by Dr. Marazzo, who will give us an update on where we stand with the development of the Holy Grail, an STI vaccine. So our first speaker is Dr. Kellum. Dr. Connie Kellum is a professor of global health, medicine, and epidemiology and director of the International Clinical Research Center at the University of Washington. She serves as faculty for University of Washington Centers for AIDS Research Behavioral Science Core, the HIV Prevention Trials Network, and the Microbicide Trials Network. Dr. Kellum is an infectious disease physician, epidemiologist, and clinical researcher by training, and her research interests focus on HIV prevention strategies, including HIV pre-exposure prophylaxis and prevention and treatment of STIs. She has led multi-center HIV prevention efficacy trials, including 2 landmark studies on genital herpes suppression for prevention of HIV acquisition, as well as HIV transmission. She co-led the Partners PrEP study, which demonstrated high efficacy of tenofovir and emtricitabine tenofovir prophylaxis, which contributed to FDA approval for PrEP for HIV prevention, which is a cornerstone in the National End the HIV Epidemic Initiative. Currently, Dr. Kellum's research focuses on implementation science research about PrEP implementation and STI interventions for young African women and men who have sex with men in the U.S. And of particular relevance to her presentation today, she recently received NIH funding for a randomized open-label study to evaluate doxycycline post-exposure prophylaxis for men who have sex with men and transgender women living with HIV and on PrEP. So I want to thank both of you for being here today, and now I'm going to turn the imaginary podium over to Dr. Kellum.
Thank you, Laura. Can you hear me okay?
Yes.
Great. So it's a privilege to be here today with you all virtually and to talk a little bit about this topic of doxycycline prophylaxis for bacterial STIs. And I'll try to summarize the rationale, the evidence, and a number of ongoing studies. Let me just see if I can get a... So as you all know, and I'm sure are hearing a lot about during this conference, is that we continue to see this ongoing increase in bacterial STIs, not just in the US, but globally. On the left is the data from WHO showing that we're close to 400 million new cases of curable STIs globally. And on the right, the CDC data suggesting that we're continuing to see rises in STIs over the last 5 years. And this is all in the context of concerns about antimicrobial resistance with gonorrhea and the observations that syphilis rates continue above, even above the rates that we were seeing before AIDS, now spreading into heterosexual networks. And we'll continue to learn about how much COVID impacts STIs and whether or not decreases are real due to behavior change or just due to decreased testing. But it's a very dynamic time in terms of STIs. Just focusing a little bit more on the data for syphilis, gonorrhea, and chlamydia in these 3 slides the numbers just show the ongoing increases over the last 5 years for each of these STIs. I think syphilis is one where we're most concerned because the rates have continued to increase, not just in MSM in the right, but also now in the last several years in heterosexual women and heterosexual men. We know that about half of primary and secondary syphilis in the US are in men who have sex with men. We also know that repeat syphilis is relatively common. And as we've begun to hear more about, we're beginning to see not only increases in primary and secondary syphilis in women, but also very disturbingly increases in congenital syphilis. So what to do and why? The why, for a moment, we believe that part of the rise in STIs is related to public health successes, actually. That as we've endorsed the U equals U concept with undetectable HIV virus being untransmittable, that may have led to relaxation of some behavior change in men who have sex with men and others infected with HIV. But also PrEP has been another potential contributor. And again, bringing a long-awaited biomedical intervention to prevent acquisition of HIV, but also maybe changing behaviors. So as with anything, there's often an upside and a downside to some of our new interventions. Last year, Jason Ong and colleagues from WHO published an excellent systematic review and meta-analysis about the global epidemiology of STIs among persons using PrEP, and I highly recommend this paper. And they did a careful collection and review of data from many parts of the world and showed overall that there appears to be about a 10-fold higher incidence of gonorrhea, chlamydia, and early syphilis in PrEP users compared to non-PrEP users attending sexual health clinics globally. Some of this, I think, is a sign of success, that we're targeting PrEP to those who potentially are also at not just high risk of acquiring STIs, but HIV. So I think we have to always remind ourselves when we see these kind of data that is the intent of PrEP, is that it should be a targeted intervention delivered to persons at high risk of STIs and HIV. Data published last year as well from Australia looked at the incidence of STIs in almost 3,000 men who have sex with men in Victoria in the PrEPX project from 2016 to 2018. And they followed persons for a little over a year on average and found the striking STI incidence of 92 per 100 person years of follow-up. Much of this was gonorrhea and chlamydia, lesser incidence of syphilis. They did analyses to find out the predictors of incident STIs and found that young age, greater number of partners, and group sex were all associated with incident STIs. And I think when we come to talk about doxycycline pre- and post-exposure prophylaxis is important. One of their important findings is that they were fairly highly concentrated, that a quarter of men in this cohort accounted for 3/4 of incident STIs, as shown in the graph on the right. So why do we care? Why should we look for new interventions? It's partly because STDs cause morbidity, particularly syphilis in both MSM and women with the risk of congenital syphilis, but also because in HIV uninfected persons, STDs increase the risk of HIV acquisition. And that's true not just for symptomatic, but asymptomatic STIs. These data, which came from a cohort in New York, showed that for MSM who are diagnosed with rectal GC or chlamydia, They had about 1 in 15 were diagnosed with HIV within a year. For those with primary or secondary syphilis, it was 1 in 18, and that's in comparison to 1 in 53 for MSM who had neither of those infections. So there's been a lot of discussion and effort to refocus our efforts in terms of doing something about this STD epidemic. And this excellent paper by Bob Eisinger, Emily Erbelding, and Tony Fauci and JID a couple of years ago highlighted 3 big areas. One is point-of-care STI diagnostics. Secondly, STI vaccines, and you'll hear about that from Dr. Marazzo in the next presentation. And then therapeutics. Clearly, we need new effective drugs for gonorrhea, but also they talk in this commentary about the potential role of antibiotics for pre and or post-exposure of STIs. So when we talk about this, we also have to anchor this discussion in the context of the need to be antimicrobial stewards and to reduce the risk of antimicrobial resistance. And drug-resistant gonorrhea is one of 5 infectious diseases that CDC considers an urgent threat. And in the last year's report, they showed that although there have been some gains in terms of antimicrobial resistance with an increased focus on antibiotic stewardship, that there still continues to be a concern about GC. So I recommend this report to summarize some of these issues. So given that we need to ideally identify new interventions in this context of an ongoing bacterial STD epidemic in some populations, we and others have considered what antibiotics could potentially be used for STI prophylaxis and settled on doxycycline. And the reasons are that it's safe, well tolerated, available generically, and is inexpensive, therefore. It's been used safely and effectively for prophylaxis of Lyme disease, which is also a spirochete, and for malaria prophylaxis. There appears to be no baseline tetracycline resistance, at least it's never been observed in Chlamydia trachomatis or T. pallidum, and that since tetracycline resistance is already high in gonorrhea and doxy isn't used for treatment, we're not really losing or compromising its use for gonorrhea. It's been used chronically in persons with acne, and it's not a common drug used to treat other infectious diseases. So it's not like we're at risk of losing an important part of the antibiotic armamentarium. In terms of past studies of STI prophylaxis, there was a small study in Los Angeles among HIV-positive men who had sex with men who had a history of syphilis. 30 of them were enrolled into doxycycline PrEP, so used daily, versus contingency management and no medication. So it was an open-label study, and they found a 73% reduction in STIs and no self-reported behavior change. It was really a pilot study, but it suggested that maybe there was some potential for doxycycline, in this case PrEP, to reduce STIs in men who have sex with men who are living with HIV. Steen did a meta-analysis of studies in female sex workers. Many of these were in Africa, and in these studies, most were using monthly azithromycin in female sex workers, and they found overall a 40 to 60% reduction in gonorrhea and similar reductions in chlamydia without evidence of risk compensation. They did not really, most of these studies did not look at antimicrobial resistance. And lastly, and I'll focus more on this one because it's, I think did stir the field, a study by Jean-Michel Molina and colleagues in France that looked at HIV-negative men who have sex with men in France who are on event-driven PrEP, and found about a 47% reduction in gonorrhea, chlamydia, or syphilis with doxycycline post-exposure prophylaxis. So just to spend a moment on this was a subset of about 232 men who were followed after the IPRGAY study showed efficacy of event-driven HIV PrEP to be randomized in a 1-to-1 ratio to doxycycline versus no doxycycline PEP. They were told to take 200 milligrams within 24 hours and no more than 72 hours after each sexual episode. They were told not to take more than 6 tablets per week. They were tested every 2 months for syphilis, gonorrhea, and chlamydia. Vast majority were white. They were in their late 30s, and most were employed and college educated. Very sexually active, an average of 10 partners in the past 2 months and 10 sex acts in the past 4 weeks. 17% had STIs at baseline, and they were followed for an average of almost 9 months. And the overall results are shown in this slide, where they found the 47% reduction in chlamydia, gonorrhea, and syphilis as a composite endpoint. And this is the Kaplan-Meier curve showing the time to the first STI, where you can see that the curves parted pretty early within the first several months of follow-up and stayed separated. So they had a pretty high incidence of STIs, 46 per 100 person years in doxy-PEP group and almost 80 per 100 person years in the no PEP group. Notably, about 70% of these STIs were asymptomatic, and overall, this was accomplished with a pretty low exposure to doxycycline, the median of 680 milligrams of doxy used per month. Relatively small proportion of people discontinued doxy due to adverse events, which were mainly GI. Just to break it down, really that overall reduction was seen only for chlamydia and syphilis. Here are the data for chlamydia. They had 28 participants who had incident chlamydia, and you can see the dramatic reduction in chlamydia in the doxy-PEP group. These are the data for syphilis, where the incidence was lower, but they also saw a 73% reduction. And these syphilis cases were primary, secondary, and early latent. And again, you see the overall reduction and that the curves stayed separated. They saw no reduction in gonorrhea overall. And this was perhaps not too surprising because the prevalence of tetracycline resistance in France in GC is about 70%. They did see lower rates of urine and anal sampling for GC-PCR, but no difference in pharyngeal. So maybe there was some suggestion of a benefit in urine and rectal GC, but not in pharyngeal. They found no difference in doxy-PEP between in risk compensation in terms of sex acts, sex partners, condomless sex. They found a suggestion to a little lower condom use over time in both arms, and they found no incident HIV. They looked and tried to do some evaluation of antimicrobial resistance, but didn't have great success in terms of getting cultures for GC or chlamydia. So in the GC cultures that were obtained, the 4 that had tetracycline resistance all happened to be in the non-doxy-PEP arm. And they had, across the 2 arms, they found a high prevalence of genotypic markers of tetracycline resistance in those that were GC PCR positive. Out of the 24 chlamydia PCR positive, they were only able to get 4 culture positive, and all were tetracycline sensitive, and they did not attempt to evaluate doxy resistance in syphilis. So it taught us some important lessons. Even with a relatively small sample size and short follow-up, it appeared to be efficacious. This was in a pretty homogeneous population where men who have sex with men were already doing event-driven dosing for HIV pre-exposure prophylaxis. They have a high prevalence of tetracycline resistance in GC and in Europe overall. They only looked in men who have sex with men who didn't have HIV, no transgender women, and limited evaluation of antimicrobial resistance. So there were a number of questions after IBRGE about whether doxy-PEP would work in persons living with HIV, in persons taking daily rather than event-driven PrEP, in younger populations, and among women, in Africa where chlamydia prevalence is quite high in those starting HIV PrEP. Would there be some benefit against gonorrhea when tetracycline resistance is lower than in France? Impact on genitalium, and what would the impact be on antimicrobial resistance? So after IBRGE, we and others did a survey of on the Grindr app and found that among the pretty diverse 1,300 participants who responded, there was very high interest in doxy-PEP, both in terms of trying it or being in a randomized trial. So there was interest. I think it also stirred up a controversy among providers about whether the risk-benefit ratio was in the favor of doxy-PEP and concerns about antimicrobial resistance. And Stephanie Cohen has the honor of having argued for doxy-PEP against Mike Cohen 2 years ago on this conference, and in July taking the no side against Basil Donovan. So a lot of debate about whether this is ready for prime time, and a real concern about whether or not, if it does work, what would the impact be on resistance in chlamydia and syphilis, and also in other organisms like MRSA, commensal Neisseria, and the gut microbiome. There's a nice review last year in CID about ongoing studies, both PEP and PrEP, and I'll highlight some of these in the next few slides. So Annie Luchtmeyer and I are co-PIs on a study in San Francisco and Seattle where we're enrolling 780 men who have sex with men and transgender women, half living with HIV, half not, and who are on PrEP, who've had a STI in the past year, gonorrhea, chlamydia, or syphilis, and we'll be doing this in the two STI clinics, Harborview and City Clinic in San Francisco and Seattle, and the two HIV clinics, Ward 86 and Madison Clinic. And it's an open label study as well, 2-to-1 randomization of PEP versus no PEP, with 200 milligrams of doxy taken within 24 to 72 hours. Participants will be followed quarterly with 12 months of follow-up. And our primary outcome is looking at STI incidence, and secondarily, we'll be doing a deeper dive into looking at antimicrobial resistance, GC, commensal Neisseria, staph, and also gut microbiome. And we're looking for a 50% reduction in the composite endpoint of time to first incident STI. And we hope that this will provide more evidence about effectiveness in MSM, particularly those using primarily daily PrEP. We'll also have data with an equal number of MSM and transgender women living with HIV. We'll learn about adherence using an app as well as hair collection, acceptability, user experiences, and we'll be looking at using molecular techniques to look at resistance in chlamydia, syphilis, as well as looking phenotypically at GC, commensal Neisseria, and staph. We, before COVID, enrolled 99 persons and fairly diverse cohort. We still are trying to get more transgender women in. The majority, we found high prevalence of STDs in the past year, part of our eligibility criteria, and we're now enrolling again. So we hope to get back on track. Study being done in Kenya is by my colleague, Dr. Jared Baton, and Elizabeth Bakuzi is looking among HIV negative women on HIV PrEP, where we found really in this and other studies very high prevalence and incidence of chlamydia in these young women where syndromic STI management has been the norm, and there's been a lot of unrecognized bacterial STIs. Since Kenya is one of the flagship countries enrolling women into PrEP, it seemed like the logical place to look at whether doxy-PEP would work there. They are also doing an open label RCT with doxy-PEP versus no PEP, and we'll also be looking not just at effectiveness, but on antimicrobial resistance in gonorrhea and chlamydia. They had enrolled 58 women prior to the COVID epidemic, and are actually continued to do some enrollments during COVID. They're getting a young population, and 23% had an STI on enrollment. There will be a study, again, by the French group under Jean-Michel Molina's leadership to look at not just doxy-PEP, but also the Bexsero meningococcal B vaccine study. This will be a factorial design looking at doxy-PEP and meningococcal B vaccine, and they are targeted to enroll 720. They have been held up by COVID, but anticipate starting in the not too distant future. Lastly, I'll just highlight there's a single-arm study that started just before COVID, looking at doxycycline PEP or PrEP, and this is really looking at acceptability. They will, in a pre-post way, look at effectiveness against STIs and safety, tolerability, and some resistance data. They're giving MSM a choice and transgender women a choice of either daily doxy PrEP or episodic PrEP. They're enrolling both persons living with HIV and not, and in the case of those who are not infected, those who will be on PrEP. So I'll just summarize and say that we have a lot to learn still. I think we need to know, is it safe? Is it effective? What are the effects on antimicrobial resistance? We'll learn that in MSM and transgender women living with HIV or on PrEP from the US, Canada, France, and Australia. We'll also learn about young heterosexual women from Kenya on HIV PrEP. And it's one of these balances of, does it work? Is it safe? Does it have an impact on antimicrobial resistance? If it does work, I think we view this as a very targeted intervention. Modeling studies from Australia showed that if we could reach that subset of people who have the highest risk of repeat STIs, that we could see 50% reduction in syphilis in a year if 50% of persons adopted it and it had 70% efficacy. So it may offer short-term intervention until STI vaccines become available. And in many ways, this is very analogous to the rationale for HIV PrEP while we were evaluating candidate and continue to evaluate candidate HIV vaccines, and where HIV PrEP has had a big impact. So I just want to thank my co-PI, Annie Luchtmeyer, and the doxy-PEP team in San Francisco and Seattle, and my colleagues, Janelle Stewart, Jared Baton, and Elizabeth Bakuzi from the Kenya DPEP team, Jean-Michel Molina, who led this initial study and will be also doing the new study, the factorial design, John Calder and colleagues from the Australia Syphilaxis study, and our funders, DMID, NIAID, and NIMH. So I'll end there, and thank you very much.
Thank you, Dr. Kellum. I'm now going to introduce our next speaker, Dr. Jeannie Marazzo. Dr. Marazzo is a professor of medicine and director of the Division of Infectious Diseases at the University of Alabama at Birmingham. She's internationally recognized for her research and education efforts in the field of STIs, especially as they affect women's health and in the setting of HIV. Dr. Marazzo is a fellow of the American College of Physicians and of the Infectious Diseases Society of America, and was elected to the IDSA Board of Directors in 2018. She has a long and storied career as relates to conducting research on the human microbiome, specifically as it relates to female reproductive tract infections, hormonal contraception, and its relationship to women's risk of STI HIV acquisition. She served as the protocol co-chair for the VOICE study and serves as the biomedical on the biomedical science committee of the Microbicides Trial Network. Even more recently, Dr. Marazzo, she's currently serving as the principal investigator for an NIH-funded phase 2 randomized observer-blind placebo-controlled study of the efficacy of the vaccine Bexsero, which is already licensed to prevent meningitis, but specifically for the efficacy against gonorrhea. In the midst of leading an infectious disease division and conducting cutting-edge research, as demonstrated by her talk yesterday, Dr. Marazzo is also heavily immersed in studies and communication around the COVID pandemic. On a fun note, not only has she become a regular on CNN and other news outlets, but she's developed quite a following for her iconic blue glasses, which we can see today. So with that, I will turn the podium over to Dr. Jeannie Marazzo. Thank you.
Laura, thanks. That's probably one of the nicest introductions I've had, although when you say long and storied career, I start to think that you may realize that I'm about to enter a new decade, but that's okay. We won't talk about that now.
Okay.
So it's super fun to be here to talk about this, especially to follow on Connie and also to be right before the ASTDA award luncheon, which is one of my favorite events, and I'm so sad I won't be able to see all of you in person. So I was asked by Laura and colleagues to talk about the current state of the STI vaccine field, and I have to disclose right off the bat that I am not an immunologist. I'm not even a vaccinologist. I am a clinical trialist with an interest in translational STI research. So please know that there are probably many people listening who could give this talk in a much more deeper way, and also who could probably correct several mistakes I'm going to make. But I do have the advantage of having I think a 30,000-foot view, and also telling you, I think, about some of the exciting aspects of the emerging clinical research on particularly Bexsero. So it won't be a deep dive, but I hope to give you some good updates, and also at the end to pose some provocative questions that ask, so if we did have an effective STI vaccine, what would we do with it, and how would we really get it out there? So what is the current status of the vaccine field? And some of this involves what the rationale is for a vaccine, which is no mystery to this audience, in which Connie just emphasized very nicely. We have exploding incidence of both bacterial and viral STIs, as well as protozoal in terms of trichomonas, antimicrobial resistance, especially in gonorrhea and M. genitalium. Our infrastructure is really strained in terms of trying to screen, diagnose, and treat most common STIs. So really, prevention is long-term the only answer. The plausibility has really shifted, and I'm going to talk to you a little bit about why this is. And if you are not really in this field, this may be the most important component of what you might learn today. And I'll tell you when that slide is queued up. There are really exciting new methods for understanding the biological basis for the mechanisms of immunity, especially for some of the pathogens that we're most familiar with, like gonorrhea, chlamydia, and to some extent Treponema pallidum. And then the third thing is that there is, I think, a commitment that is finally really taking hold. There's always been a strong commitment in the STI field to vaccine, of course, really started with HPV and its success. But in the last 10 years, this has consolidated largely under the leadership of several people at WHO, Sammy Gottlieb, CDC, and Carolyn Deal at the NIH. And you can see there that there have been several really nice consultations that I'll reference throughout. And also, the WHO has actually proceeded to define some preferred product characteristics for specific vaccines. They already have a document for herpes simplex, which I show there, which is a really interesting read, and they are in the process of doing a similar document for gonorrhea. And then finally, again, under Dr. Deal's leadership, the NIH, NIAID refunded the STI Cooperative Research Centers in 2018, and those are now fully focused on vaccine development for chlamydia, syphilis, and gonorrhea in the basic and translational science realm. So it's a really exciting time, and there are several things that I think will hopefully convince you that we have come a long way since Sammy Gottlieb gave us this graphic of the state of the vaccine landscape in 2016. And you can see at that time, we had really only one vaccine sort of pathogen focus that was all the way down to clinical evaluation, and that was herpes. And we definitely have come a long way. I'm going to revisit this at the end and ask you where you think we are. I'm going to ask whoever's not muted to mute their line because I can actually hear some stuff. So I'm going to start with gonorrhea first, because that's really where we are farthest right now. And it's a fascinating pathogen to think about vaccination for. And one of the challenges in figuring out how to become or get someone to become immune to gonorrhea is that humans have no protective memory response. And how do you know this? You know this because as clinicians and public health people, you see people with recurrent gonorrhea constantly. I think it was Kim Warkowski who told me she had a patient who had something like 50 episodes of gonorrhea in her clinic. I'll have to ask her for the accurate number. You also can see reinfection with the same strain and the same serotype. So it's not a matter of the pathogen taking advantage of shifting its shape in order to evade the immune system. However, there were some old clues to the fact that maybe you do make some immunity to this pathogen. In fact, came from early work by Frank Plummer and colleagues in women in South Africa, looking at the risk of upper tract infection as a function of production of bactericidal antibodies. And Mike Cohen and others with their human model pursued this line of research, and that has really given birth to a whole field. I'm not going to take you through this very dense slide again. I'm going to refer to a couple of really nice compendium reviews that I did review for this talk, not to drag you through the details, but just to point out that there are several excellent documents that can tell you exactly where we are in the quest for vaccines for these pathogens. And I like to show this one because it's part of several articles in a nice Frontiers in Immunology compendium. And this is from Ann Jurst, Michael Russell, and Scott D. Gray-Owen, who have been at the forefront of gonococcal vaccine research, particularly in the murine model now, for several years. And they basically take you through the objectives and advantages and then some comments about each of the models that have been used to develop a gonorrhea vaccine, including the naturally infected human model. So it's very helpful, but again, we're not going to go into detail because I want to sort of keep us focused at the higher level picture, and I want to quickly talk to you about some of these other exciting projects. So let's continue on with gonorrhea, and let's remind ourselves why we have thought that the meningococcal group B vaccine in particular might afford an option to protect against gonorrhea. So we've known, as we have increasingly been able to characterize the outer membrane vesicle, or OMV, of Neisseria gonorrhea, that it's really, really homologous, very similar to the same structure in Neisseria meningitidis. For those of you who don't know that bug, Neisseria meningitidis is a bacteria that can cause fatal spinal meningitis. There are several serotypes that are most common. And we've had a vaccine to 4 of those, A, C, Y, and W, for a long time. We have not had a vaccine for group B until relatively recently, when Bexsero and another 1 or 2 actually came out. And there are some complex reasons for that. It's been a challenge, and I'm going to talk a little bit about how that puzzle was cracked, because it gets to, I think, helping us understand how we're getting further along the path to developing some of these vaccines that we really need. So there was a theoretical basis, knowing how similar this antigenic component of Neisseria meningitidis and gonorrhea were, the outer membrane vesicle. And there had also been some hints in vaccine campaigns that had targeted group B meningococcus, particularly in Cuba and in Norway, that there was maybe an association with a concomitant subsequent reduction in incident gonorrhea. But it had been difficult to precisely pinpoint until we saw this really beautiful paper, a study led by Helen Patusis Harris's group in New Zealand. And what they did, as many of you know, but I'll just review quickly, was to take advantage of a gigantic natural experiment in a country with excellent sexual health records and excellent healthcare, as well as a relatively defined small population, as we know from their beautiful ability to largely contain COVID-19. So what happened in New Zealand was that there was an outbreak of group B meningitis with Neisseria meningitidis in the 1990s, and people were enrolled in a vaccination campaign to prevent that potentially fatal infection. So a lot of people got vaccinated with this group B New Zealand meningococcal vaccine. These investigators had the brilliant idea of using the subsequent decades of sexual health records to say, let's go back and look at whether the people who got that vaccine were more likely or less likely to develop gonorrhea in our system over the next 20 years. Well, really 10 years, a little bit later. At 20 years, actually, because it was from the vaccination started, as you can see, with the green there in the late 80s and went through. So they did this, and they were able to look in this way at the incidence of gonorrhea in these people by their vaccination status. And they also did a very cool thing, which was to look at the incidence of chlamydia, reasoning that comparing chlamydia and gonorrhea would give you some sort of similarity in terms of STD risk, but obviously the meningitis vaccine was not going to protect you against chlamydia. And what you can see here, so on the left-hand side, you've got vaccination status of participants by their year of birth. And in green, these are people who were partly vaccinated. And in blue, you can see as people were born in the mid-80s and went into the 90s, they were more likely to get vaccinated against the group V meningococcal vaccine. And what you can see over here are the number of gonorrhea cases that occurred in the vaccinated versus unvaccinated people in green versus blue over the time period of 2004 to 2014, and compare that to chlamydia. And the bottom line is that the meningococcal vaccine reduced the incidence of gonorrhea by about 33%, and there was no reduction either in incidence of chlamydia or gonorrhea or chlamydia co-infection. So this was a really exciting finding, and if you turn that into an estimated vaccine efficacy with adjusted odds ratios, adjusted for ethnicity, sex, age group, socioeconomic status, and geographic location, they were still able to show a fairly significant reduction by 1/3. So that's really the background. So what does that mean? And I want to bring you back again to one of these articles, compendium articles that Sammy Gottlieb, Ann Jurst, Carolyn Deal, and others wrote just last year in Sexual Health that now takes a more global view of the overview of gonococcal vaccine R&D. And again, this gives you a sense of what the strategy or candidates are and the status and results. So again, this is a little bit of a reference article for you to know to go to. But the exciting thing about this whole area, and this is the slide that I hope those of you who don't really do this stuff very much remember, is that none of this really would have happened without something called reverse vaccinology. And for those of you who are at the ISSTDR meeting in Vancouver, it seems like a billion years ago now, but it was just, I think, a year ago, last July, the opening plenary was given by Rino Rappuoli. And Rino was the originator of the effort with his team to define appropriate antigens for Neisseria meningitidis that would make up ultimately Bexsero. And he used this approach called reverse vaccinology. And at this point, that version was sort of version 1.0, and I'm going to explain what it is. So what you do in reverse vaccinology is basically turn the traditional laboratory approach to defining antigens, the things that provoke your immune system to make antibodies, that we used to use in the laboratory on its head. So when you grow a pathogen in the laboratory, whether it's chlamydia, whether it's gonorrhea, or whether it's a virus, you identify various components of it and you hypothesize which of those components are going to be most important for defining or provoking your immune response. And that has worked. But it's also been associated with some pretty spectacular lack of efficacy trials, which I may mention in a moment. What you do in reverse vaccinology is actually start with the genetic sequence of the pathogen, and you do what's called whole genome sequencing. So you're basically looking at all of the genes in a given pathogen. And you use that approach with bioinformatics to discover candidate antigens that are likely to induce protective responses. And so on the right-hand side, you can see a summary of how the Bexsero antigens were generated using this approach. And so there were almost over 2,000 candidates predicted by whole genome sequencing in the sequence of Neisseria meningitidis. They then went down to identify those that were exposed on the surface of the bacteria, and that cut out a whole bunch of them, so you only had 650. They then went down to express, purify, and immunize mice with 350 of them. Again, went back to see which of those were surface exposed, so mice most likely to encounter the human immune cells during infection. And then they showed which of those induced bactericidal antibodies in that model. And they were able to select 3 antigens that fit all of these criteria really well. And those are listed here. I'll tell you what they are in a moment. It was incredibly exciting and really new. And then the new version of reverse vaccinology, which is beautifully described by Reno in that YouTube video when he won the Gardner Prize in Canada just a couple of years ago. I encourage you to watch it if you're interested in it. Takes this a step further, and what that does is deeply sequence genes that encode variable regions of the antibodies in a given B cell repertoire. And what you can do then is allow for cloning of those antibodies from patients who may be getting better, particularly from viral infections. And why do you know about this now? Because there are gigantic studies from Regeneron and Eli Lilly looking at coronavirus monoclonal antibodies as treatment and prevention. So you're basically giving these antibodies to people to mimic natural antibody combinations in a host. And the cool thing about these is that you can actually combine more than one for a cocktail of antibodies. And that's exactly what some of these companies are doing with COVID. So it's a very cool technology that you should know about. So here's where we landed with Bexsero, which is also called 4C meninge B. It is like a super version of the New Zealand strain of this vaccine. So again, the New Zealand epidemic strain that you saw had a 30% reduction, had the old outer membrane vesicle antigen that I mentioned. But this one also has 3 of those purified proteins listed here that have different functions in the bacteria and that were identified with that reverse vaccinology approach. So the hope is that if we saw a 30% reduction with the straightforward New Zealand vaccine, perhaps the addition of these 3 potent immune response driving antigens will actually give us a higher efficacy. And that is indeed what we are hoping to demonstrate with the MAGI study, which is a phase 2 randomized observer blind placebo-controlled study looking at the efficacy of this vaccine in preventing gonococcal infection. And this is a study that we are leading. It's sponsored by the National Institutes of Allergy and Infectious Diseases, working closely with Carolyn Deal, Lori Newman, and others there. And we are doing this in the structure of the NIH-funded STI clinical trials group, in support with GSK and also the Uniformed Services University of Health Sciences. There'll be several sites here in the US, including Emory, LSU, and UAB, as well as facilities associated with the USUHS in the Washington area. And then we'll also have 3 sites in Bangkok. We're planning to enroll 2,200 participants. And the primary objective, of course, is to look at prevention of urogenital and or anal rectal gonococcal infection. We are going to look at pharyngeal infection, and I don't have time to go into that, but that could be the subject of another talk, because remember, Neisseria meningitidis really does infect the nasopharynx. That's how it gets into your body. That's one of the places that it's colonized. It's a necessary colonizer there, although we know it can cause urethritis too. So it's going to be really interesting to see what happens with, I think, oropharyngeal colonization with this product. And of course, we're interested in safety, although this is a very safe vaccine out there in the world. And of course, we will be looking at neutralizing antibody induced by this vaccine by collecting PBMCs in a subset of patients. So that's all I'm going to say about gonorrhea, which I did want to talk about for most of the talk, because that is really close. In fact, we are on the verge of getting single IRB approval. We have gone through almost all of the necessary steps at FDA and NIH, and we anticipate enrolling our first participants, assuming COVID permits that, hopefully sometime in the next maybe 6 weeks. So fingers crossed, we will let you know when that happens. Just a few slides on some of the other pathogens, because they are really not as far along, although there have been some very, very hopeful developments. What about chlamydia? Well, we do know that using animal models, particularly mouse models, that you can induce at least short-lived natural immunity to this pathogen. And in fact, Will Geisler has shown very nicely in a cohort of women here in Birmingham that in women who cleared their infections spontaneously between tests and treatment, they were less likely to become reinfected during follow-up, even though they were just as likely to be as sexually active as their counterparts who did get reinfected. And that suggested that there was a mechanism associated with the ability to clear spontaneously and protection against recurrent infection. That said, we know that we need, as we do probably for many of these pathogens, a combination of protective mechanisms, including antibodies, but very important tissue resident interferon gamma producing CD4 positive T cells. And that's something that's been really another holy grail in looking for chlamydia vaccine candidates. And again, this is a slide from Dr. Geisler, very kindly provided this to me. It's a summary of the preclinical pipeline of the products that are being looked at, and generally they fall into one of three categories. It may be a subunit vaccine based on the membrane, the major outer membrane protein of chlamydia. You can do a whole inactivated vaccine, and there are also live attenuated plasma deficient vaccines in process. A couple of years ago, actually, it was just 1 year ago. It's also very hard to believe it was just a year and maybe almost 2 years ago now. This publication in Lancet Infectious Disease prompted a really nice media event. Actually, people were quite interested. It's a small phase 1 study of a candidate that is a recombinant engineered version of that major outer membrane protein, and it's combined with a potent adjuvant. And it was a first in humans phase 1 study, 15 women. So this is really important that they used women in my mind, because women are the ones who are going to benefit most from a vaccine given the upper reproductive tract sequelae of chlamydia trachomatis. And they looked at it, interestingly, by giving it intramuscularly for 3 doses, and then they gave 2 intranasal boosts. And the reason for giving intranasal boosts was to try to stimulate mucosal antibody, reasoning that cervicovaginal antibody would be the first line of defense against any STD, right? Acquired genitally, anyway, as opposed to other ways. And it was really interesting. They showed that in the group that got the active adjuvant and the vaccine candidate, that they induced over 4-fold serum IgG seroconversion in almost all, and they were able to detect vaginal immunoglobulin as well. And very interestingly, the amount of vaginal immunoglobulin correlated with the serum immunoglobulin. So this was really hopeful. It is being pursued, and I think it was kind of the first large signal in humans anyway, that something could potentially be done. This would be a major impact on women's reproductive health. Unfortunately, with herpes vaccine, a lot of work has been done in 2 areas, prophylactic, and that's not unfortunate that the work has been done. It's just that we're not as far along as I wish we were, and I think many in the herpes field wish that we were. 2 categories, prophylactic and therapeutic vaccines. Prophylactic vaccines, I would say, from my read, took up much of the last couple of decades, and that's giving vaccine to people who have never been infected with the herpes virus, genital herpes viruses 1 and 2, but primarily 2. Most of these vaccines have targeted the surface glycoproteins GD2 and GB2. One of those, of course, is the one that we base our diagnostic testing on. And they all have been promising in animal models, and even though they elicit neutralizing antibody, they tend not to have worked in clinical trials, and it's been a bit of a disappointment. And I just want to remind you of this really incredibly well done study that, again, is one of these studies that people refer to as a failure, which of course makes me crazy, because we learned a huge amount from this very well done study. And I think it's really important not to forget this was a gigantic study, over 8,000 sexually active women who were seronegative for HSV1 and 2. They got a GD2D subunit vaccine. Or they got a hepatitis A vaccine, which was kind of a cool control. And this did not work for prevention of HSV2. And they defined HSV2 incidence either by seroconversion or clinically confirmed, culture confirmed clinical disease. But interestingly, there was a 35% reduction of vaccine efficacy for HSV1, and they showed that higher antibody titers to HSV1 was associated with vaccine efficacy. So I would say that this study had a huge role in informing the plausibility of the field going forward and in our understanding of exactly where to go with it. The other side of the coin for HSV2 is therapeutic vaccines, and those have really taken off in the last, I would say, 5 to 7 years. And in these studies, you are vaccinating someone who's already infected with HSV2, and you're looking at whether or not the vaccine can reduce your rate of shedding in particular. So the idea would be to make you less infectious, even if you are already infected with the HSV2 vaccine. And there's a lot of really nice things about these designs because you can compare the participant to themselves pre and post shedding rates after immunization. And you can, and that gives you a lot more latitude in terms of the numbers of people you need. And just some of the candidates that have been or might be studied depending on COVID are listed there. Again, don't have time to go into the details, but it is an area that I think still does hold promise. The last sort of content slide for pathogen specifics is just to tell you that Treponema pallidum has been a very tough nut to crack. I will say one of the fun things about doing these talks is when I try to refresh my images. And when I went to look for a new Treponema pallidum image, I realized that you could get a cell phone cover with Treponema pallidum. So I'm sure that Sheila Lukart has one of these, which makes me jealous. But I do love that phone cover. So speaking of Dr. Lukart, she worked with Jim Miller as a graduate student, and we've known for a long time that you can actually immunize rabbits with irradiated T. pallidum and protect them from reinfection. The challenge with Treponema pallidum is that it has very labile, unstable, indeed, surface antigens, right? So you're not ever looking at the same bug, basically, when you go from person to person. Really unique surface. It doesn't have a lot of outer membrane proteins like the MOMP of chlamydia that you can target, and it's very fatty, which makes it really tough. So current efforts, again, no time to really focus on these, but they are now using this reverse vaccinology approach. One of the CRCs that I mentioned is focused on this and working very hard to identify some functional approaches to identify these antigens. And I think it's going to be a really exciting field in the next 5 years. So just in the last couple of slides, I'll try to take you back to what really happened. So what if we find that Bexsero works? What if we find that this exciting chlamydia vaccine candidate goes into phase 2 trials? It's always this question of efficacy to effectiveness. So efficacy is what you see in a clinical trial. Effectiveness is what you see in the real world. And the best example I think of this is HPV vaccine in clinical trials. HPV vaccine is pretty much perfect, right? It prevents pretty much everything that HPV can do. But what's the challenge with HPV vaccine? It's implementation. And we know that even before COVID, we had challenges scaling up in parts of the world where cervical cancer still kills women in an alarming proportion. And I just yesterday saw this nice commentary by Melissa Gilkey and colleagues talking about not just getting HPV vaccine on track in terms of general implementation, but dealing with the shortage that happened last year and now dealing with a disruption by COVID. So challenges are real. Also, vaccine hesitancy, right? Major barrier, huge measles outbreak as shown there last year. And unfortunately, in the COVID world, lots of people talking about mistrust. Especially exacerbated by this warp speed concept, which some people love the term, some people don't love the term because it just makes them more suspicious that we're not going to do this safely and well. And then I guess the last question that I have for you all to think about is if we do have an effective vaccine, not an efficacious vaccine, do we really have the political, economic, operational climate and the commitment to create mass opportunities for an STI vaccine that we can't masquerade, quote unquote, or market as a cancer vaccine. An HPV vaccine is not masquerading as a cancer vaccine. It is one, but we certainly have tried to market it as one. We really can't do that with gonorrhea. We maybe can do it with chlamydia a little bit by saying it's a women's health vaccine, a reproductive health vaccine, but that's also sidestepping the reality that we need STI vaccines. People have sex, people need sexual health protection. And so I think we need to embrace this and just call it what it is. And I would end by just pointing out that Julie Silver's talk yesterday really rung true for me, talking about how to talk to people about having safe, quote unquote, sex during COVID. And really, it's about sort of embracing the debate about vaccination, about STIs, about sexual shame and stigma. And I just wanted to remind you that some of the people who have taken on the quote unquote anti-vax movement, also really more, I think, professionally called vaccine hesitancy, have a pretty cool campaign that I first learned about by seeing on a bumper sticker called Vaccinate Your Crotch Goblins. And it's basically a Facebook page and a very large advocacy organization to try to really talk to parents who are reluctant to vaccinate their kids and to just confront fears, anxieties, concerns head on, and with humor, I think, and with acceptance. And I think, as Julie said, that's really the only way we're going to get to getting an implementation plan for these vaccines. So I want to really acknowledge several people who very kindly either gave me slides or whose work I relied on very heavily. Will, in particular, Christine Johnston, Sammy Gottlieb, Lori Newman, and Caroline Cameron. And I will stop there and thank you very much for your attention. Have people already left? So I can't hear anyone, and I'm not sure what's going on. Looks like Laura's online. I could just, Connie, I don't know if you're still here, but if Laura is offline, I don't think, yeah, I don't think we have time for questions. Laura, you're coming back online. It looks like there you are. But I think that Christine Johnston's still here. Excellent. Okay, I am going to just thank Connie for a really great talk. Thank Laura for convening us. And I think I am supposed to ask people to go to the ASTDA luncheon. And I think that's it. Connie, did you have any final words of wisdom?
No, I loved your talk. Thank you. It was
Yeah,
fun to do this together. And
absolutely.
complementary approaches. Hopefully, we'll see a STD vaccine and look forward to the data from the Bexaro trial.
Okay, thanks everybody. We'll go to the ASTD luncheon and talk soon. Bye.